This is the humble cell, a building block of life. Much happens within this little cell at an incredible pace to convert DNA into proteins. The blueprints are the DNA. Instructions are carried by the messenger RNA to be transcribed at the ribosome. These instructions are interpreted by the transfer RNA. Modifying this interpretation process opens doors to a world of possibilities, which is called tRNA engineering. The field of tRNA engineering is about incorporating all kinds of new genetic functions via the tRNA. This is used for synthesis of drugs, to produce nanomaterials, to track proteins and build new peptide databases. It allows the creation of new and novel organisms. The opportunities are significant, and you can also see why this emerging field would have ethical concerns as well. A world with tRNA engineering will introduce this dynamic field by starting with the role tRNA plays in translation, then highlighting some of the new and modified translational machinery and how this works. We'll talk through some technical specifics to give you a deeper understanding before spending time on recent applications. tRNA engineering in its simplest form is engineering of the translation machinery, and therefore a method to manipulate the genetic code. We use the transfer RNA for the delivery of a unique and new amino acid known as an unnatural amino acid to the mRNA that is busy producing peptide chains in the ribosome factory. You can think of this tRNA as being a spaceship, but instead of soil from Earth, it is now carrying soil from Mars and delivering it to Earth to create something never seen before. Next is a refresh of what happens in the natural process, and just think that it works pretty much the same from a big picture view. It is the small details that change.
The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain.
So what changes? And how is tRNA engineered in the first place? Amino acyl transfer RNA, or charged tRNA, meaning it has an amino acid chemically bonded ready to be used in the translation process, like we saw at the start of the video, can be genetically modified to deliver and bind to an unnatural amino acid, thus delivering them onto the growing peptide chain to produce new or novel proteins. There are 20 amino acids occurring in nature. The engineering of new pairings of orthogonal amino acyl tRNA synthetase with the tRNA molecule means that 70 unnatural amino acids have been added to the genetic code so far. These have a wide range of useful structures and functions. It has been done in E. coli, yeast, and mammalian cells. This breaks the rules of the genetic code, providing genetic code expansion. So how does an unnatural amino acid get incorporated? Unnatural amino acids are assigned specific codons, which are either a stop codon or a dedicated codon previously occupied by another natural amino acid. The use of what is referred to as a mischarged unnatural amino acid tRNA pairing allows the addition of the unnatural amino acid at the special codon that is matched by the anticodon of the tRNA. This uses a modified amino acyl tRNA synthetase to charge a particular unnatural amino acid onto a specific tRNA molecule. Then these pre-charged unnatural amino acids can be introduced into the peptide chain via the mRNA. Some of these techniques are more developed than others, and we will talk specifically about 2 techniques. The stop codon method that uses sense codons and stop codons to place the unnatural amino acid, and the orthogonal method that involves entirely engineered and unique amino acyl tRNA synthetase and tRNA pairs. You've been watching the stop codon method. Here is an application where the stop codon technique is used in combination with an orthogonal pair. This uses a genetic incorporation of a D amino acid into green fluorescent protein. D amino acids are widely distributed in living organisms. In order to be able to incorporate them into the fluorescent protein, they need a specialized tRNA synthetase that would work in combination with a connected specific tRNA molecule. This is known as an orthogonal pair in that it provides a totally unique combination that will only carry the D amino acid required and will not accidentally pick up a similar natural amino acid. For this, they construct an orthogonal pair using a plasmid that contains 2 copies of the engineered amino acyl tRNA synthetase gene and a copy of the suppressor tRNA gene. Then they make a change to the codon that will incorporate the new D amino acid and change it to a stop codon. Now they have the orthogonal machinery ready to incorporate a new D amino acid. Tyrosine 66 participates in the formation of the fluorophore, which is what makes the protein fluoresce green. Making changes to this residue will alter the spectral properties of the protein, so they decide to choose an aromatic amino acid L-phenylalanine to replace tyrosine 66 and create a mutant as a control. They then make the D-phenylalanine mutant once again, replacing tyrosine 66, and introduce a stop codon just for this residue. This is translated into the green fluorescent protein using the new orthogonal machinery in E. coli. The mutant, containing D-phenylalanine, was compared to the control. To their surprise, they saw the configuration was stable and saw an obvious red shift in the fluorescence. Its emission and excitation maxima had changed. This meant there was a change in the backbone of the protein, and D-phenylalanine was incorporated successfully. The thermal stability of the fluorescent protein was shown to improve as well, a very useful application in research to track metabolic progress. The second application for microbes is even more radical: the utilization of DNA with 6 bases instead of the 4 known bases, accompanied with a modification of a tRNA molecule that can recognize the new base, allow the engineering of a new bacterium. Its ribosome uses unnatural bases, DNAM and DTPT3, to insert an unnatural amino acid into a growing protein chain, allowing cells to synthesize both natural and unnatural nucleic acids. The genome of every cell on Earth uses 4 DNA bases: adenine, thymine, cytosine, and guanine to encode proteins. Scientists have long dreamed of expanding that set to create cells that work with both natural and unnatural nucleic acids. An expanded collection of bases allows cells to synthesize unnatural amino acids to produce proteins that have new functions. A strain of E. coli was created to work with DNAM and DTPT3, the 2 aromatic DNA bases that pair with each other through complementary packing and hydrophobic forces. This is instead of the usual hydrogen bonding that natural bases use. They engineered the bacteria with a circular DNA plasmid that had a gene for green fluorescent protein. Inside this gene, they inserted the unnatural base pair at a site where they wanted to incorporate an unnatural amino acid. They tested the system with 2 different tRNAs, each carrying a different unnatural amino acid, an N-propagyl lysine and a P-azidophenylalanine, and successfully demonstrated that the bacteria added each at the proper spot in the green fluorescent protein. The work shows that unnatural base pairs are compatible with the molecular biological machinery inside cells. Huge. This changes what has always been available in nature by manipulating it in laboratories, expanding its potential, and creating a new era of genetic alphabet expansion. Alongside technical issues that come with any emerging field of engineering that in time will no doubt be resolved, we come back to a core issue of safety. What happens when these genetically modified organisms integrate into nature? If they are used for drugs such as vaccines, Are we ingesting or injecting foreign particles into the human body that can cause other unexpected issues? tRNA engineering is allowing a new bold future to unfold rapidly with both opportunities and risk, and it is for us to choose the applications wisely.